So we're moving on to four short talks now.
So it seems if you do full screen, it doesn't auto-advance, but slideshow it does.
Okay. And I'm sitting there and I'll give you 1 minute warning.
Right. Hi, everyone. I'm Saket. I'm a PhD candidate at University of Southern California. I want to start off with thanking the organizers, both ISCB and OBF and BOSC, for giving me a chance to talk about my package, pysradb, which is a package for downloading, querying metadata and data from NCBI Sequence Read Archive. NCBI Sequence Read Archive is a repository of next-generation sequencing datasets. It has a collection of all the public datasets that exist out there, which have followed next-generation sequencing. The corresponding the corresponding repository for gene, which is specific to gene expression datasets, is called GEO. These datasets have been made. These repositories have been made it possible for us to test any novel hypothesis, and they serve as a test bed for reproducing any analysis that has been done in the literature. However, there are a few problems with getting both the metadata and the data. Accessing this data and metadata is not as easy as it should be. So while developing a database for a PhD project that I'm working on, I encountered few key problems that hindered obtaining both data and metadata from these two resources. I'll walk you through what those problems are. The first problem that you face is getting the metadata. This metadata is required because any sample or any sequencing that was done has a corresponding label attached to it. It could reflect what condition that particular sample was sequenced on. This is done right now through NCBI's web interface. So you go onto a web page, you download the run info table, and you get a sequence. You get a text file which has all the metadata related to it. Okay. Imagine doing this for hundreds of thousands of projects. If you're building a database which has multiple data sets, doing this manual process is going to take ages. The second problem that comes while accessing this data is because just to give you an overview as to how the data is organized, the data sets at SRA are hosted in terms of projects. Each project has multiple experiments. These experiments are reflective of what condition a particular sample was sequenced in. And each experiment, which is referred by here as SRX, has multiple runs associated with it. It could have one run, it could have multiple runs associated with it, which are basically reflective of technical replicates in most cases. So in order to be able to replicate the entire analysis of the entire project, you want all these files to be accessible. Right now, that's made possible by using FastQDump. It's a tool made available through SRA tools by NCBI itself. FastQDump requires accession IDs, which are the run IDs here, the SRRs. So now again, in order to be able to replicate the entire analysis, you want to be able to download all the files. However, since FastQDump requires accession IDs, you do not maintain this hierarchy of the project that you require downstream. Okay? And the third problem comes because of the interpolation that you require between GEO and SRA. So any data set that gets submitted to GEO, which is specific to expression datasets, has a corresponding SRA ID associated with it. In order to be able to download the raw data associated with this GEO project, you need the corresponding map to the SRA project, which is given by the SRP ID here. Okay? In terms of the current problem, in order to be able to find out what that corresponding project is, you again need to go to the GEO website and find out what particular project it pertains to. So in order to be able to address all these problems, I came up with this tool, which is called pysradb. Pysradb basically gives you a seamless access into both downloading the metadata and the data from NCBI's Sequence Read Archive. It is designed so that you do not require any programming background. Everything is built up as a command line tool. Both metadata retrieval and data retrieval happens through command lines. So I'll start off with solutions for examples. So the first example is getting the metadata. You can just type on pysradb metadata and gives you the metadata for an entire project. The second thing that happens is the second problem that I addressed was about getting the data. So in principle, you can just do pysradb download, and it downloads the entire project rather than downloading individual files. I just also want to point out it supports the recent change that has happened in SRA, where SRA is now moving to Google Cloud Storage rather than FTP. In terms of the third problem, it also allows interpolation between SRA and Geo, so you can convert Geo ID into SRA ID on your command line. This is published in F1000 Research, so I also want to use this opportunity to thank the reviewers, Simon Andrews and Ryan K. Dale. And finally, I'm presenting a poster on this today. It's poster PO1. It's available freely. The tool is available freely on GitHub. It's licensed PST. It's also available as a package on Bioconda. And I also want to use this opportunity to thank this travel. My travel would not have been possible had it not been the award from Open Bioinformatics Foundation and also ISCB's travel award. Thank you.
So we're collecting questions for the next 4 talks to a question and answer session afterwards. Another short talk coming now.